Hi everyone. Today, we're going to examine a study that puts forward a novel strategy for addressing age-related diabetes. The research investigates whether small vesicles from stem cells can rejuvenate senescent pancreatic beta cells, the cells responsible for insulin production. In this explainer, we'll break down the methodology, the key findings, and the precise molecular pathway the researchers have uncovered, which could be quite significant for developing future treatments. So here's our roadmap. We'll start by establishing the context of beta cell senescence and its role in aging. Then, we'll look at the evidence showing how these stem cell-derived vesicles appear to reverse that process. From there, we'll dissect the mechanism, identifying the specific molecule doing the work, the signaling pathway it targets, and how that all ties back to rescuing mitochondrial function. We'll then conclude by summarizing the findings and their therapeutic implications.
So, let's start with the central problem. As we age, the pancreatic beta cells that produce our insulin can enter a state of decline, a process we call senescence. This cellular aging is understood to be a major driver of type 2 diabetes, primarily because these senescent cells lose their ability to secrete insulin effectively when glucose levels rise.
To be more specific, senescence is a state of what's thought to be irreversible growth arrest, usually triggered by cellular stress. For a beta cell, this means it stops proliferating. But more importantly, it fails at its core function, secreting insulin. On top of that, these cells adopt what's known as the senescence-associated secretory phenotype, or SASP. This means they start releasing pro-inflammatory factors that can disrupt the local islet environment and contribute to systemic insulin resistance, which only makes the problem worse. Now, the current therapeutic approach to this has been focused on drugs called senolytics. Their job is pretty straightforward. They selectively eliminate these senescent cells. And while this can be effective, it also means you're reducing the total mass of beta cells, which raises some potential long-term safety concerns. The strategy being explored in this paper is fundamentally different. Instead of just getting rid of the damaged cells, the goal here is to rejuvenate them, to reverse the senescent phenotype and restore their function, all while preserving the overall cell population.
So, the question is, does this rejuvenating approach actually work? Let's turn to the core data from the study, starting with their in vitro experiments.
This figure summarizes the key results from their cell culture models. The researchers took beta cells, induced senescence with oxidative stress, and then treated them with vesicles derived from human amniotic mesenchymal stem cells. The main finding is that the treatment worked. As you can see from the data, classic molecular markers of cellular aging and DNA damage were significantly reduced. At the same time, the cells regained their ability to proliferate, and the expression of genes tied to senescence was suppressed.
Of course, reversing molecular markers is one thing, but restoring function is what really matters. And this figure shows that function was indeed restored. The treated beta cells regained their capacity to secrete insulin in response to a glucose challenge. It also appears to have corrected underlying metabolic defects. The mitochondria in these cells resumed more normal function, which was measured by oxygen consumption rates and ATP production, and they also generated fewer damaging reactive oxygen species.
Now for the crucial next step. Does this hold up in a living organism? Here, the researchers turned to a mouse model of age-related diabetes. After a systemic injection of the vesicles, they saw a significant and sustained improvement in metabolism. The treated mice had lower fasting blood glucose, and as the tolerance tests show, they also had improved glucose disposal and better insulin sensitivity. This points to a real systemic benefit.
To confirm where this metabolic improvement was coming from, they performed a histological analysis of the pancreas from these mice. What they found was that the islets of Langerhans from the treated animals showed far fewer signs of cellular senescence. Key aging markers were down, and importantly, the expression of master regulators of mature beta cell function, namely PDX1 and MAFA, was restored. This gives us direct evidence that the beta cells themselves were being rejuvenated in vivo.
Okay, so we've seen evidence that these vesicles work, both in cell culture and in a living animal. The next logical question is how? What exactly inside these vesicles is mediating this rejuvenating effect?
Remember, these vesicles contain a complex mix of proteins, lipids, and nucleic acids. So the next task for the researchers was to pinpoint the specific active ingredient responsible for the anti-senescence activity they were observing.
The investigation homed in on a specific microRNA, MIR21-5P, as the key effector. To validate its role, they performed two critical experiments in mice. First, they administered a synthetic mimic of this microRNA and found it reproduced many of the same therapeutic benefits as the complete vesicles.
Second, they did the opposite. They used a specific inhibitor to block MIR-21-5P's function within the vesicles before injection. In that case, the therapeutic effect was significantly blunted. Together, these experiments establish MIR-21-5P as a necessary and at least partially sufficient component.
All right, so if MIR-21-5P is the key molecule, how is it having such a profound effect inside the beta cell? What is its direct molecular target? To identify the target, the team started with a transcriptomic analysis to see what genes changed expression after treatment. This pointed them toward a pro-inflammatory pathway involving the cytokine interleukin-6. Using computational algorithms, they predicted that MIR-21-5P could bind directly to the messenger RNA of the IL-6 receptor alpha subunit, IL-6RA, and they confirmed this prediction with a dual luciferase reporter assay, which showed that MIR-21-5P does in fact bind to the IL-6RA transcript and inhibit its translation. So, by reducing receptor levels, it effectively dampens that entire downstream signaling cascade. This brings us to the final piece of the mechanism. How does inhibiting this one inflammatory pathway result in a complete rescue of mitochondrial function? The researchers were able to trace the pathway to its final effector. They used genomic mapping techniques to identify the binding sites of the key downstream signaling protein, STAT3. This revealed that in senescent cells, activated STAT3 was binding to the promoter region of a critical gene called MCU, which encodes the mitochondrial calcium uniporter, and this binding was shown to be actively repressing MCU transcription.
So, to put that plainly, in senescent beta cells, you have elevated IL-6 signaling. This leads to the phosphorylation and activation of the STAT3 protein. Activated STAT3 then moves into the nucleus and binds directly to the MCU gene, effectively shutting down its expression. Since MCU forms the primary channel for calcium to get into the mitochondria, a process that's essential for ATP production, this transcriptional repression by STAT3 provides a very direct link between the inflammatory signaling and the mitochondrial dysfunction seen in these cells.
To confirm this last link in the chain, the researchers performed a crucial rescue experiment. They treated senescent cells with the beneficial MIR-21-5P, but at the same time, they used RNA interference to specifically block the expression of MCU. The result was that the therapeutic effects of the microRNA were almost completely abolished. The cells stayed senescent, and their mitochondrial function remained poor. This confirms that restoring MCU function is the critical downstream event in this rejuvenation pathway.
Okay, let's take a step back and put all of these findings together to visualize the complete signaling pathway that this study has mapped out.
So what we have is a complete and coherent signaling cascade. It all starts with the delivery of MIM-21-5P by the stem cell vesicles. This microRNA then acts as a direct inhibitor of the IL-6 receptor, reducing its expression. This dampens the inflammatory signal, which leads to decreased activation of STAT3. With STAT3 no longer active, its transcriptional repression of the MCU gene is lifted. MCU expression comes back online, allowing calcium to flow into the mitochondria. This in turn reboots mitochondrial function, reverses the senescence phenotype, and ultimately restores glucose-stimulated insulin secretion.
As the authors conclude, this work presents a novel xenotherapeutic strategy, one that's based on rejuvenation, not cell elimination. By uncovering this very specific molecular pathway, from a single microRNA in a vesicle to an inflammatory receptor to a transcription factor, and finally to a specific mitochondrial channel, this research provides a new mechanistic blueprint for developing therapies that could potentially reverse the cellular damage that underlies age-related diabetes.